Hello, I'm Sang-Woon Lee from Gyeongsang National University in Jinju, Korea. So today I'm going. First, of course, I would like to thank the organizers for inviting me to this wonderful symposium with a variety of topics in theoretical biophysics. I'm really happy to present my network community-based analysis on human chromosome conformation data. So there are many previous or the ongoing precious collaborators of mine listed here, and some bunch of papers, and because the approach I'm going to present eventually is the result of the combination of two kind of separate series of my work. So first, I'll mainly present my work on human chromosome conformation networks analyzed with this network community detection, in particular across various scales, as the title suggests. And the second part is about more on this methodology part, the methodology on community detection itself, especially on its consistency of the result. And eventually, I'll conclude my talk with the recent ongoing work, which is basically the combination of the two topics I mentioned. So I'll start to talk about the first work by introducing the human chromosome structure. And as most of you may know, the chromosomes have this complicated, non-trivial folded structure inside the cell nuclei. So when I first learned about this structure in, for example, biology class, I believed all of these details about this packing structure is well known. But of course, it was not. And I think this is one of the many examples that are unknown even if we decoded the human genome sequence itself. So deciphering this structure is of course crucial to understand how this fundamental genetic material or chromatin works in cells. And a major breakthrough step to reveal the physical proximity between a genomic sequence is this interaction map of chromosome loci called HiC. So this HiC technology provides the physical proximity or the interaction frequency between two chromosome loci in this matrix form. And the technology itself has recently improved enormously for a couple of years. For example, the resolution of this map was in the order of megabase pair in 2009, but it was possible to get this map in the order of a kilobase pair resolution in 2014. In other words, the resolution had become like 1,000 times better during the 5 years. For comparison, well, this is not quite a fair comparison, but the resolution of the screen of iPhone in 2009 and that of the iPhone in 2014 are less than 5 times difference. Well, it's not quite a fair comparison. But anyway, you can see some non-trivial structures here, like some block diagonal and this some off-diagonal structures here. And this kind of structure is, in fact, an important clue to reveal the 3D structure. So, for instance, these off-diagonal part in higher resolution is the evidence of the loop structure, as the title of this paper suggests. And another famous example in the low resolution is this block substructure called topologically associated domain, or TAD, which obviously represent the local packing, or in terms of this network structure composed of loci and as a node and the weight as a weighted edges. To me, it looks like a typical structure of this community structure in network, which is a famous concept in the network science. So let me introduce this community structure in network first. A community structure of a network refers to this kind of a division of the entire network into these two pieces, for example here, where each of them is statistically more densely connected inside than the inter-community connections. And this is a fairly intuitive concept, but at the same time quite practical to see the structural and or functional subunit lurking in a network system. For the interested audience, I can recommend some review papers listed below here, and already published more than a decade ago now. So And one of the most conventional ways to detect such a community structure is to maximize this objective function called modularity. And basically, it quantifies how many of the edges are intra-connected one compared to the background or the expected ones expressed by these null model terms for a given community partition. I'll come back to this null model part soon in the context of a chromatin interaction. And so maximizing this function corresponds to finding the most densely intra-connected subpart. And by the way, I always emphasize the sort of control parameter here is this Kronecker delta term here, embracing the community division, because this part is in fact already given when network structure is given, but this Kronecker delta term determined which part is actually a count. So a bit rather technical but important part you should remember throughout my talk is this resolution parameter gamma in front of this null model term. And it controls the overall scales of the communities found, where the larger value of gamma represent the smaller communities or larger number of communities as illustrated here. And the problem of maximizing the modularity is, of course, it's quite computationally challenging because of the combinatorially exploding numbers of possible divisions here. And in fact, so naturally, the how to cleverly found some approximate solution is the key to this modularity-based technique. And as I told you, we need to talk about this null model term here, especially for this chromosome interaction data. And this if you look at this term, this product of the degree or weight here assumes the only information available to us is the strength of background interaction for each node. So we use this best guess that the interaction between the two nodes will be proportional to those background interaction. However, in the case of the chromatin data, we know much more than that here. The additional information is, of course, the fact that the chromosomes are basically 3D folded structure constructed from the 1D sequence. In other words, we can clearly distinguish between the cases of the pairs of loci located far away from those very closely located in the genomic or 1D sequence. Moreover, thanks to the studies on this contact probability scaling as a function of sequence distance, we know that the background interaction between the two loci follows this so-called fractal globule scaling, with the contact probability will be expected to be proportional, inversely proportional to the 1D sequence distance. So we use this so-called fractal globule null model term, composed of both the overall interaction term, represented by this product of the Ki and Kj. On top of that, we consider this the decaying rate of this interaction following the 1D structure itself. So we use this kind of specially tailor-made null model term throughout my work. For the audience technical enough in this field, we use the version 2.1 of the GenLuma algorithm for all of the results I present from now on, which is a variant of the famous Lubang algorithm, where the nodes are greedily merged to maximize to the modularity. And due to the stochastic nature of merging, it is inherently stochastic, which would be actually the key aspect in the second part of my talk, so I'll just briefly mention it here. Before we jump into the real high-CMAP data, we test our community detection algorithm with various scales on the model structure called conformation dependent polymerization or CDP fractal globule model here, which supposedly mimic the chromatin packing with the fractal globule scaling. And here I have to thank Young-Hoon Kim and Postec who generated this CDP model and cross-checked the results with me. And with this example, I would like to emphasize a very important merit or the strong point of our algorithm compared with other conventional definition of TED, which is this ability to detect these non-contiguous communities, like this segment, A, B, C, and A, B again. So in addition, as a result of the different resolution parameters are all independent to each other, the communities across different scales are not necessarily strictly hierarchical. So in fact, many conventional TED detection methods impose such a strictly hierarchical restriction. So our method is, well, quite flexible and versatile in terms of expressional power. So I'll show you the examples of such a versatility for the high-C map data later. But for now, I'll provide some evidence of validity of our method with the fractal global model here. So the best part of using this kind of model system is, of course, we actually know the exact detailed structure, the 3D structure of this model system. So let's see if our community detection actually successfully find the compact structure of this model polymer system. So the plot on the left shows that the 3D distance versus the 1D or sequence distance inside the detected community. And as you can see clearly here, the 3D or Euclidean distance between intra-community node pairs is systematically shorter than the random counterpart. For example, the distance for the randomly chosen the two node in the case of equilibrium or the case of equilibrium globe model, both for intra-community nodes and randomly chosen node. And this clearly distinct scaling behavior for the CDP model's intra-community structure hold for the rescaled the 3D distance divided by the radius of gyration as well. So it means that our community detection indeed identifies loops or at least a compact substructure. And in addition to such a scaling behavior, we measure geometrical properties such as the asphericity or the relative shape anisotropy. So we can check the communities we detect here for the CDP model are, again, systematically take more compact or literally the globular structure. So based on this validation with the polymer model system, we move to the real high-C data of the chromosome chromatin interactions. So we use this particular high-C map data introduced in this Cell paper in 2014 with the 100 kilobase pair resolution and converted this raw data to the normalized version of the interaction with this particular normalization scheme for some technical audience. By the way, this original Cell paper itself provides the set of, I have to emphasize, the single resolution TED data. So we used that as some kind of reference later. So here's the representative result of the communities we found with our method for 3 different resolution, gamma equal to 0.5 and 0.6 and 0.7 for the chromosome 1. And here are some, in this part, here are some examples of the non-contiguous communities. I mean, these yellow pieces in fact constitute the same community, this yellow one and this light blue pieces. And of course, we can detect communities for all of the other chromosomes like this. And by the way, the black stripes here are represent the non-coding region, by the way. And as I already introduced, although many communities in different resolution looks like being almost like a nested, but not exactly not strictly true, as you can see from this part. And unlike the polymer model system, with the real 3D structure of the chromatin interactions is of course not known, so we have to rely on some relevant biological factors for verification, of course. And the first signal is this well-known biological marker called CTCF, which is supposedly enriched in near the chromatin domain boundaries. So we compare this CTCF enrichment near the community boundaries from our method with the TED from this kind of a standard data provided by this Cell paper. So if there are significant overlap like this, the it means that our method can detect chromatin domain boundaries, just like the data provided by this cell paper. And of course, in contrast to the single resolution TED, our method can detect the communities across various scales. So depending on the resolution parameter gamma here, the overlap between community boundaries and the CTCF enrichment is, of course, this changed or fluctuated. And as you can see here, for chromosome 1, around gamma value close to 0.7, the overlap between the community boundary points we found and both the CTCF enrichment and this TED boundary point reported in the cell paper become maximum, measured by this precision value. Therefore, if we adjust the resolution parameter properly, we can recover this TED with the property of the CTCF enrichment as a biological evidence. And as you can see here, the number of TED or the number of our definition of the community boundary or TED or non-contiguous pieces of community per community has these peaks around gamma value close to 0.7 again here. So we suggest the TEDs are kind of the most fragmented community, I mean, if after normalization, as you can see from the fragmented colors here, in this example here. So we successfully recovered this TED structure at this particular range of gamma. Then what about the other ranges of gammas or resolution? So one of the earliest findings about this chromatin interaction is this separation, so-called A and B compartment, using the simple PCA reported in this science paper in 2009. And if you detect the, by the way, this A-B compartment has this property of the one of them is genetically more active than the other. I kind of forgot which one is which, but it's not important. But if you detect the communities for very low resolution or with the very low values of gamma, can you recover this A-B compartment separation as well? And it's of course possible in principle because the A-B compartment here presented in this paper are non-contiguous checkerboard patterns, and our method, I mean, in principle, can detect such a checkerboard pattern as well. So, which is something that cannot be detected with the many conventional TED finding method I have to emphasize. So here's the evidence of the recovery of A-B compartment from our method. So it's not just the in principle possibility, but it's in practice it actually works. If we tune the parameter range low enough, then we obtain two communities. And as you can see from this overlap table here, the largest community from our method roughly correspond to the B compartment, and the second largest community roughly correspond to the A compartment. So if I summarize the result reported in the paper published 2 years ago, by tuning the resolution parameter in the community detection scheme, we could recover previously reported substructures of the IC map based on this two separate verification. So we kind of suggest, in this paper, we suggest the whole spectrum of community scales that may be meaningful or for different biological contexts. And recently, in collaboration with the great collaborators in Umeå again, we started to analyze this semi-hierarchical or bow-tie structures of these high-C communities more deeply. So hopefully I can report about this result of this exciting ongoing work soon. So for the rest of my talk, I will return to a bit more technical aspect about the network community detection problem itself, which is about this typical dilemma of the community detection, nicely expressed by Professor Hang-Yeon Jo at Catholic University of Korea, an APCTBA alumni, by the way, in response to this Peter Holm's more general statement on the modeling studies on Twitter. So if we know the real answer, in our case provided by the biological factors, what's the point about this detecting community? And if we don't know the answer, how can you test the validity of the detected community? So from now on, I actually replied to him by advertising our work. So from now on, I'll talk about our attempt to tackle this issue based on the concept of inconsistency or the self-consistency. So let's go back to the square one of the community detection problem. So this kind of statement, detect the community structure, looks like a completely innocent order if you just study network community detection. If you are a practitioner of community detection, or if you try to actually find communities with a stochastic algorithm, you will immediately encounter this kind of situation. So suppose you run this algorithm and get this nice result. Well, that's it. This is a community structure, and you may think that you can start to write a paper. But unfortunately, if you run this code again and again, you will get these results. In other words, you will end up with this ensemble of community structure, not a single one by a stochastic algorithm. So the issue here is the inconsistency of the result. And so is this necessarily a bad thing that should be kind of fixed? Well, as we can use, in fact, we kind of turn the table by using this inconsistency to study some kind of a structural property of the network. So first, we can construct this so-called co-occurrence matrix for each node pair, whose element indicate the fraction of the node pair belonging to the same community in the ensemble of these community structures found. So if a pair of nodes always belong to the same community or always belong to a different community, the matrix element would be 1 or 0, like this, respectively. So the intermediate values here, corresponding to this, the intermediate node, indicate the inconsistency. So in this example, only this 3 node here, obviously the 3 node between the two obviously defined bulk of communities are responsible for this inconsistency. So based on this co-occurrence matrix, we formulated the inconsistency called companionship inconsistency, or COI, of the individual node by simply symmetrizing it and taking the average over all the other nodes. So we can treat this COI value as a new kind of a node centrality to quantify the local measure of community inconsistency, which may eventually reveal a functional flexibility of the node, which is of course important in some biological context as well. So, and if you are familiar with some node centrality in network science, you may ask, this kind of bridginess can be detected with the well-known centrality called betweenness centrality as well. But here's our counter example. Oh, I forgot to bring the counter example, sorry. But due to the time limit, I'll skip the part. You can check our paper for the detailed comparison of this. And so as an appropriate example, we generate the clustered Erdős-Renyi random network, where we loosely connected the pre-assigned community composed of the dense random network and put a number of inconsistent node here intentionally. And the pre-assigned community structure is colored accordingly on the left side, and the shade on the right side correspond to this compendium inconsistency value. So if we just try to detect the community structures here, the central part composed of this a bunch of inconsistent nodes is labeled just randomly, essentially. And the inconsistency measure effectively capture this capture the fact that membership is very inconsistency in this central part. So you cannot trust the labels here. And as some of you might know, this is the most famous or infamous example of this network community, the network community called Jackary's Karate Club network, where Karate Club was split into 2 communities as a result of the fight between the instructor and the administrator, reported by this sociologist Wayne Jaccary in the 70s, which is the very first example the network scientists always tackle with the newly developed community detection method. And at this particular value of the resolution parameter gamma, we found this interesting node here with the absolutely larger value of companionship inconsistency than any other node, meaning that the member is most neutral about this reported dispute inside. Especially because this node has only two neighbors, we call this kind of outsider. And the second example is a slightly bigger one, the Star Wars character network. So for those of you who know this stuff, these are the networks of characters appearing in the original trilogy of the movie Star Wars. And they are connected if they appear in the same scene. So in this network, the Luke Skywalker, the main protagonist of the entire series, naturally interact with many characters in the different communities, including his father. Sorry for the spoiler for this 40-year-old movie, but of course he's far from being outsider because he's a main protagonist. So we call him kind of a multiplayer characterized by this large degree and large companionship inconsistency value simultaneously. So we can, for example, set up some ad hoc measures like a product of the degree and this companionship inconsistency value to characterize this multiplayer, distinguished from the outsiders I mentioned in the karate club network, just as an example. In case you are not comfortable with our results based on this famous or infamous karate club characters or karate club network and fictional characters, here are more real or real world examples network. For example, if you look at the inconsistency value of this Chilean power grid network, these nodes have show more gradual changes of companionship inconsistency values across different nodes, which may imply different levels of hierarchical community structure. And I'll come back to this exact point at the later. So in the paper published 2 years ago in collaboration with Professor Hitae Kim at Caltech in Naju now, we presented the main idea of using a local community inconsistency as a kind of a nodal centrality and its implications, and of course the correlation between the inconsistency and other centrality measure. But we believe that there should be more systematic studies on this problem. First, we took the resolution parameters here rather arbitrarily, and we didn't have this kind of a global measure to quantify the overall network-wise inconsistency. And thankfully, Taekyung Lee, a smart and outstanding graduate student back then, read our paper and contacted us for the potential follow-up study. So it was a perfect timing for us. And Taekyung came up with the global measure of inconsistency called this partition inconsistency, or pi, based on the cutting-edge similarity measure for different network partition, and more sophisticated local measure of inconsistency called the membership inconsistency, MEI. Sorry for coming up with lots of abbreviation. And both of which have this nice scaling property, namely the effective number of different configurations in the global level and the independent membership in the local level. So for details, please check our recently published paper. I will introduce shortly. And instead of going through all the mathematical terms here, here is an illustrative example to show the difference between the MEI and KOI. And here I demonstrate the newly developed MEI measure is good at detecting the kind of a bulk part of the inconsistent regions, while the KOI is good at the boundary of even the boundary of the inconsistent regions fundamentally, because it's a maximum at the region where the node switching between just the two community. But in this case, I show this 3 community case, so different local inconsistency measure deals with the situation differently. And by far, the most important aspect of this recent work is that we finally started to look at the whole spectrum of community by adjusting the resolution parameter, the gamma. So by looking at the global measure of inconsistency, or pi, as a function of community resolution, we can pinpoint the ranges of gamma where the inconsistency become very small, or in other words, consistent, or reliable communities are found, especially when the number of communities stay at this flat integer value, just like the shaded regions here. And by using this series of model network with the single level and double level or hierarchical communities, we verify that our method can find this most consistent community across different community resolutions. So single resolution here and the double ranges of resolution here as exactly expected. And by taking various real-world networks again, we can actually pinpoint the regions of the meaningful community structures. So if you recall my assumption of the hierarchical community structures in the Chilean power grid here, it is actually verified here by observing this stepping structure, very nice, clean stepping structures of the number of communities with very low values of inconsistency or the consistent or reliable community across different scales. So I mean, it's not quite easy to get that clear-cut evidence in the real network, as many network scientists, if the audience would agree. So for all of the details, please check our recently published paper. So with this inconsistency analysis, we suggest to pinpoint the most reliable ranges of community scales in the high-CMAP data I mentioned in the first part of my talk. So in collaboration with the Umeo group again, I got this preliminary result that there are actually several regions of gamma with this consistent community, which may correspond to our previous rediscovery of this AB compartment and the topologically associated domain, or TAD, and potentially suggest some other types of biologically unexplored areas. So we are working on this and hopefully get some meaningful results soon. So the most important aspect is that thanks to our inconsistency analysis, it's possible to narrow down the most relevant ranges of gamma even without external biological information. So let's see how it goes, and hopefully I can report the results soon as well. And of course, for the record, I didn't mean to undermine the importance of biological factors in any way, especially in this symposium, as all of you will agree. But I present this inconsistent analysis as another kind of augmentation of our series of community detection approach. So here's a summary of my talk. In the first part of my talk, I introduced our network community detection scheme to detect the communities in the high-C data or chromatin interaction across different scales. And in the second part, I introduced a bit technical but practical issues of community inconsistency problems, and how we can use this inconsistency information itself to quantify the reliability of the community detection across different scales is an important topic. And finally, as an ongoing work, I present this work in this overlapping part by the combining the two, the high-C data analysis in terms of community inconsistency to narrow down the meaningful ranges of gamma, which is especially important in this biological data with many unknown pieces. So thank you for listening, and of course I have to thank all of my collaborators again, especially the two of the organizers of this symposium, Professor Jae-young Jeon and Professor Ludwig Lizana, because their joint initiative with this Korea-Sweden cooperation grant a few years ago was the beginning of my journey on this topic. So if you have any question about anything, please send me emails to this address. And as a final note, the pandemic is not over yet, as all of you know. And so stay safe, wear a mask, and get vaccinated if you can. I mean, I got my booster shot last weekend, so I can meet all of you in person more safely in not too distant future, just as in this great in-person meeting at Kiruna in January 2019. Thank you. So one example I presented is this AB compartment here. So I forgot to mention, but this the separation of this entire genome sequence into the two pieces in a given chromosome by to A and B, and they are actually this checkerboard pattern. So it's not like they just cut the half somewhere in between, and the left one is A and right one is B. But it's based on this principal component analysis. They put the positive part as A and the negative part as B. So the fact that our method is versatile or flexible means that we can detect all the way from this very coarse level of separation in the very low resolution, and this very fine-grained resolution as well. I mean, if we just slightly tune the gamma parameter from 0.5 to 0.7, we have this huge different scales of the communities here. So this AB pattern is definitely one example of these non-contiguous communities. I'm not sure if I answered to the question. So the number of trial, I guess you mean this kind of 10. I mean, 10 is of course too small. I mean, it's just to illustrate the examples here. But yeah, that's a very good question. And if I remember correctly, most of these results are from 100 trials, or if Daekyung is in the audience, maybe he can answer better answer. But we haven't actually systematically studied those dependency on the reliability of this reliability. So that's a very helpful comment that we should be coming. Oh yeah. And of course we are aware of that problem, because this the community inconsistency or the modularity landscape is another huge topic in this area. So yeah, we'll need to think about this more. Actually, Daekyung left the answer in the chat window. It's usually 100 to 500 was enough number, but I mean, it can depending on the modularity landscape and the number of nodes, or I mean the situation in general. Thank you.